Hello, welcome to this Science on Sundays talk. I'm Beverley Glover, I'm Director of the Cambridge University Botanic Garden, and I'm going to try and give you the Science on Sundays talk that we would normally be giving in the garden in April 2020, here in this recorded format. The Science on Sundays series is about introducing our visitors at the Botanic Garden to the work that scientists do using the living collection we maintain. It's a very informal talk series. It lets our visitors find out about some of the science that happens using the plants, and I'm going to try in this recorded talk to keep to the same informal style. For those of you who know the Science on Sunday series and have come to the talks before, I hope this informal style will be welcome and familiar. For those of you joining me for the first time for one of these talks, I hope you'll understand the nature of the series this isn't going to be a perfectly polished video presentation, but I hope that you enjoy it anyway. And enjoy the opportunity to learn a little bit about the science we do at the Botanic Garden. So the talk I'm going to give today is about some work we've been doing in my research group to try and understand plant speciation and how it is that one plant species evolves into 2 and 3 and 4 so that we end up with the enormous diversity of flowering plants that we see today. Now, normally I'd be giving this talk in the Botanic Gardens classroom, and we'd have the opportunity to walk around and see some of the amazing plants that are there as well. Here's a lovely shot of our bee borders, which I guess the bees are enjoying today, even though there aren't any people there to enjoy them. But one of the things as a scientist I get out of regularly being in the Botanic Garden is the sense of that great diversity of the flowering plants, of just how many different sorts of flowering plants there are. Let me explain what I mean by that by showing you a family tree of the plant kingdom. So here we have a family tree, and on this family tree I've marked all the major groups of plants. So for instance, down here at the bottom where I'm pointing now, there are some green algae, and then all of the other groups, the liverworts, the hornworts, the mosses and so on, these are all groups of plants that live on the land, and I've included some pictures of perhaps some of the less familiar ones like the liverworts. Now this family tree of plants tells you that there are only about 10 or so major groups of plants. But the interesting question for me as a scientist is about the different numbers of species that we find in each one of those groups. So now I've shown you how many species we find in each group, and you can see that there's a really big asymmetry. The flowering plants, the angiosperms up here at the top of the family tree, are very, very species rich. There are 350,000 species or so of angiosperms. All the rest of the groups are much more species poor. There are only 700 conifers, 12,000 ferns, and that's 7,000 liverworts hidden by the little video of me down there at the bottom. So why is it that the flowering plants are so different from all the other groups of plants in terms of the number of species that they have? The answer to that question comes in part from thinking about how different types of plants achieve something we call reproductive isolation. Now, reproductive isolation is perhaps a bit of an idea that you haven't come across before, so let me explain what I mean. I mean that if you want to create 2 species from one group of plants or animals, a single species, what you need to do is somehow break that group up into 2 separate groups, which are reproductively isolated from one another. So the members of group A can breed with each other, and the members of group B can breed with each other, but what they can't do is breed between groups A and B. And that can be achieved by, for instance, a mountain range coming up between the two groups. Physical ways of making it happen are perhaps the easiest ones to think about. So that reproductive isolation is the first step of speciation. Once you have 2 groups of organisms that can't breed together anymore, then over time they'll get more and more different from each other and become separate species. But it's very difficult for plants to achieve reproductive isolation. Let me show you why. So here's a hillside full of conifers, and you might remember from my family tree that the conifers are very species poor. There were only about 700 species of conifers. Now, if we wanted to generate 2 different species of conifers out of this mass of individuals, let's say divided as I'm drawing here along the line of the valley, we would need to separate the plants so that the plants on the left-hand side of the picture here were all reproductively isolated from the plants on the right-hand side of the picture here. So we had 2 separate groups, and we didn't have any breeding across that valley in the middle. But it's very hard to achieve that because of the way that conifers reproduce. What they do is that on a windy day, they throw all their pollen up in the air, just like you see in the slide here. All that pollen's being thrown around into the air, and the wind carries it to other trees, and it randomly fertilizes other trees. And so there's no control over who mates with who, and it's very difficult to imagine a scenario in which you could achieve that kind of reproductive isolation. It's very hard for plants to do. Now, animals all living together in the same place achieve it by having behaviours or morphologies that allow them to recognise members of the same species, and those behaviours and morphologies produce reproductive isolation even when the animals are all living together. So these birds are using their morphology, the blue feet, and their behaviour, this dance that they're doing, to recognise each other as part of the same species and ensure they don't reproduce, they don't mate with other birds of other species in the same environment. Plants, of course, don't have behaviour in the same way, but the flowering plants, the angiosperms, have harnessed animals as part of their reproductive strategy. And in doing so, they've been able to introduce new ways of reproductive isolation. Now, this all happens because of the use of animals as pollinators for flowers. So the flowering plants are the only plants that produce flowers. And because the flowers are attractive to animal pollinators, and those animals all vary in morphology and size and behaviour and preference, then that generates a whole diversity of selective pressures, allowing the flowering plants to become reproductively isolated into groups that are specialised on different sorts of animal pollinators. Now, understanding that relationship between plants and their pollinators is the major focus of my research group. And we use an approach that we call evolutionary developmental biology, or EVO-DEVO for short. And what EVO-DEVO does is it thinks firstly about how the traits that you're interested in are built or developed. So that's the developmental part of the picture down here. We try to work out how flowers are made. It tries to understand how those traits function. So that's the picture here with the bees. We try and understand how they affect pollinator behaviour. And then it also tries to understand their evolution. And that's the part of the picture at the top here, where we look at how those traits change across evolutionary time. And in doing all that, we hope to get a big picture of how the traits work and how they've evolved. So I'm just going to show you a couple of examples of how reproductive isolation is generated by this reliance on animals as pollinators. And I'm going to use beetles and bees as my two examples. So I've got a couple of pictures here that I've borrowed from the internet of beetles, and you can see that the beetles, very nice animals as they are quite challenging for flowers to work with. So the beetle in the picture on the left here has its mouth parts on show, that's these features here, and you can see that they're not very long, it's not a long tongue, and that they're parallel to the body. It's quite hard for the beetle to actually access food resources. And so flowers that beetles can access can't be complicated shapes or have long tubes. They have to be quite simple, flat structures. Beetles also have very poor colour vision, but quite a good sense of smell. And so a flower that's attractive to a beetle is going to be one that doesn't spend a lot of energy or effort making complicated colours and patterns, but one instead that has good scent to it. So flowers like this water lily or this magnolia are the sorts of things that are attractive to beetles. They're simple, flat structures that it's easy for those simple mouthparts to work in, and they have not very complicated colours, but often a very pleasant smell. Bees, on the other hand, are quite different. They have a long proboscis, which they can dip into flowers, and so flowers that are attractive to bees can be much different shapes, have long tubes or spurs. They can also be quite complicated and difficult to handle because bees are generally much more intelligent and are able to process complex tasks. And they also have very good colour vision. So bee-pollinated flowers are often brightly coloured. They might have different patterns of colours that attract bees or direct them to the nectar reward. Classic examples of bee-pollinated flowers might include things like the foxglove here on the left, where those lovely speckles running up the throat lead the bee to the nectar reward at the base of the flower. Or here on the right, you can see flowers that are bilaterally symmetrical, where the pollen-presenting anthers here are at the top. This is depositing pollen on the back of the bee's neck in a place that's very difficult for the bee to groom off and feed to the offspring. So flower shapes and colours and patterns tend to be much more complicated when it's bees that are the pollinator than when it's beetles. And if you can imagine those differences extrapolated across all of the hundreds of thousands of different types of insects out there and all the other animals that can do pollination as well, you start to understand where this idea of reproductive isolation through pollinator preference comes from. Now, in my lab, we're interested in all of the traits that make flowers attractive to different animal pollinators. So we think about flower symmetry and colour, shape and size, texture and so on. But I've just chosen one trait to focus on today, which is particularly relevant, we think, in reproductive isolation and in generating multiple species from a single species. And that trait is the nectar spur. And the nectar spur is the structure you can see at the back of the flower here, this extension of the petal which holds the nectar. The work I'm going to tell you about has been done primarily by a postdoc, Mario Mazwakos, and a PhD student, Erin Cullen, working together in my lab. The work is usually done by postdocs and PhD students in the research groups, and the supervisor, that's me, gets the nice job of telling you all about it. And they've been looking at how nectar spurs work to generate speciation. And nectar spurs have been described as a key innovation. I'm going to explain what that means in a minute, but I want to just show you what a nectar spur is. So 3 different types of flowers here, all with lovely nectar spurs. Here's columbine, aquilegia, you might grow these in your garden, and it has many nectar spurs per flower, 1 from each petal. You can see them here, and the nectar is held at the base of them. The orchid here in the middle has extremely long nectar spurs. This is Angrecum, it was known as Darwin's orchid, because it was Darwin who first proposed that there'd be a moth with a tongue long enough to access the nectar right down there at the base of the spur. And then top right, the flower that I'm actually going to talk about today, this is a toadflax, a Linaria species, and again we've got a single long nectar spur with the nectar held here at the base. So what do we mean by the idea of a key innovation? Well, it's a phrase that's been defined as an evolutionary change in a trait that is causally linked to increased diversification in the resulting clade. So the idea is that if you evolve nectar spurs, then all of the species descended from that individual species that evolved a nectar spur should be radiating much more quickly. So we should get more species in a group with nectar spurs than in a group without nectar spurs. That's the idea of a key innovation. Now there are 2 ways of thinking about how nectar spurs could achieve that. The conventional one is known as Darwin's co-evolutionary race. And the idea here is that nectar spurs grow in length in flowers over evolutionary time, as do pollinator tongue lengths. So this is a little bit complicated, but I'm going to try and walk you through the diagram. So if we start at the left here, we've got a pollinator, has a nice long tongue, and can just about access the nectar at the base of the spur there. You can see it's just dipping into the yellow. But here's another pollinator, the same species, and it's got a longer tongue, just like some of us are taller than others. And as a result, it can get to more of the nectar. So selective pressure is going to result in that pollinator being fitter, because it's going to get more food, than this pollinator. It's likely to leave more offspring, and in the next generation, all of the pollinators will have long tongues, or more of them. Now, at the same time, there's variation in the length of the nectar spur. So if you look on the right-hand side of the diagram, this flower's got a shorter nectar spur, and this flower's got a longer nectar spur. If the pollinator, it's the long-tongued pollinator in this case, comes to the long nectar spurred flower, this one here, then in order to get all the way down to the bottom and get all the nectar, it has to push its head right up against the reproductive organs of the flower. That's the red dot there under my pointer. And that means it'll get coated in pollen and the flower will get well pollinated. But when the pollinator visits the shorter spurred flower, it can hover a little further away, it doesn't contact the reproductive organs, and that flower isn't going to get as well pollinated. So again, we have a fitness difference. This flower will be less fit than this flower. This flower will set more seed, and in the next generation, the flowers will all have long spurs. So selective pressure favours long spurs, and selective pressure favours long tongues, and over time, the result of that is that both tongues and spurs increase in length, and so you end up with something like that Darwin's orchid. So that's the co-evolutionary race idea. Now, the other way that nectar spurs could be generating speciation and that function as a key innovation is through a different hypothesis. This is called the pollinator shift hypothesis. And the idea is that you start with one pollinator, one tongue length, one flower, and that somewhere in the species' range, the flower's range, another animal turns up that's got a longer tongue, and it's also feeding off this flower. Now this, as we can see, is a terribly inefficient situation for the flower because the pollinator isn't contacting the pollen at all. It's not going to result in very much seed set for the flower. So the flower is under enormous selective pressure to develop a longer nectar spur. That happens, and so now we have a long-spurred version of the flower, but we still have the original short-spurred version too, because the original pollinator, this purple one here, is also still perfectly effective. So where we had one type of flower, we now have two types of flower, a long-spurred one and a short-spurred one, pollinated by two different species, and therefore reproductively isolated from each other. And over time, these two flowers could become sufficiently different that they become two separate species. That's the basis of the key innovation idea for nectar spurs and how they could generate more species diversity. So we've been working with a group of plants called the ANTIRRHINEAE, and that's a tribe in the family Plantagenaceae that's probably familiar to most people watching this talk from species like the garden snapdragon, the ANTIRRHINEAE here I'm showing you, or the toadflaxes, the lineareas here, again, quite common garden flowers. Now within the ANTIRRHINEAE there are several genera of plants that do have nectar spurs, and then lots more genera of plants that don't have nectar spurs. So the green line here is dividing those up, and on the left of my green line all of these groups don't have a nectar spur, and on the right hand side all of these groups do have a nectar spur. So we've been studying the ANTIRRHINEAE and using them to ask three questions. Firstly, to test the idea of the nectar spur as a key innovation. Does it really generate greater speciation rates? Secondly, to think about how quickly nectar spurs evolve. Do they grow slowly over evolutionary time, or can they change quickly? And thirdly, to think about the developmental basis of spur length evolution. What are the things that change inside the plant that allow spurs to grow longer or shorter in different species? So, in order to test the idea that nectar spurs are a key innovation, the first thing we had to do was build a family tree of the ANTIRRHINEAE, and that's what I'm showing you here. So we've got a family tree with all of those different types of ANTIRRHINEAE species mapped onto it, and we did this by sequencing parts of their genome and then by working out the differences between the DNA of the different plants so we could produce this family tree. What I've done is colour-coded it so that groups in blue, like this one here and here, are groups that don't produce a nectar spur, and groups in red, like the linearia shown here, are groups that do produce a nectar spur. And you can see from this that there are 4 times that nectar spurs have evolved in the group. They've evolved here at number 1, here at number 2, here at number 3, and here at number 4. Having generated that family tree, we can then ask whether there are any differences between the groups in blue that don't have a spur and the groups in red that do have a spur in terms of speciation rate. And this slide is just showing you 3 different ways which we can use to assess that, to analyse speciation rate in those 2 different groups with spur and without spur. So starting with the one on the left over here, what we have is the probability of particular speciation rates for the blue group, the ones without spurs, and the red group, the ones with spurs. And you can see that the probability of a higher speciation rate is higher for the red group, the ones with nectar spurs. Similarly, in this middle analysis here, we've looked at total diversification rate in the group without spurs and in the group with spurs, and it's higher for the group with spurs than it is for the group without. And in the third analysis on the right-hand side here, we've looked at diversification rates over time, and in the groups without a spur, it's lower, it's higher in the ones with a spur. So all of these different analyses show us that actually having a nectar spur helps you speciate quicker, it generates more species more quickly, and it fits with the idea of the nectar spur as a key innovation. And the next thing we wanted to ask was how quickly do nectar spurs evolve? To do this, we took our family tree again, the one I've showed you before, and we focused in on the very most diverse part of that tree, the genus Linearia down here shown in red. And within Linearia, we focused on this group of 8 species that are found in the Iberian Peninsula. And they have a great deal of variation in nectar spur length, from about 3 millimeters in length in this species here, up to about 14 millimeters in length in this one. And we asked, how are these 8 species related to one another? Do nectar spurs evolve gradually from short to long, or are there more rapid jumps? Here's the family tree that we produced of those 8 species, and the thing I really wanted to point out here is that the shortest spurred species, that's Linaria clementii with this very short nectar spur, is actually most closely related to the longest spurred species, Linaria beckeri, with this very long nectar spur. So between these 2 species, there's very rapid evolution of spur length. It appears that spur lengths can change very quickly in evolutionary time. So if that's the case, we want to know how they change so quickly. And we've been studying just those 2 species, the sister species of Linaria clementii and Linaria beckeri, to try and understand how their spurs end up such different lengths. So here are the 2 flowers in more detail. You can see Linaria beckeri on the left with that very long nectar spur, Linaria clementii on the right, a much shorter spur, that's it here, very much smaller, but otherwise a very similar flower. The thing that was different, particularly between Linaria clementii and Linaria beckeri, was the rate of growth. And so if you look at the left-hand graph here, we have spur length against time, and it's growing much more quickly in beckeri than in clementii. And if we actually turn that into a growth rate, a change in length per day, then again the growth rate in Linaria beckeri is much higher than the growth rate in Linaria clementii. And we wanted to look more closely and find out why the nectar spur is growing more quickly in the long-spurred species. And there are 2 things that could be causing that to happen. One is that it could be that there are more cells in the longer nectar spur, and the other is that the cells in the nectar spur could themselves each be longer. So you could have more cells, but the cells are the same size, or you could have the same number of cells, but the cells are longer, the cells are different sizes. So what you're looking at here is an analysis of cell length in the 2 species across 5 developmental stages. So we're starting at an early stage, and then as the spur gets longer and longer, and you can see that there's a little bit of a difference. At the very end of growth, the cells in Linaria beckeri, that's the long spurred species, are a bit longer than the cells in Linaria clementii, but not very much. But when we look at the number of cells over time, that's really very different. So in Linaria beckeri, we have this big jump here to lots and lots of cells in the nectar spur, but we don't have that jump at all in Linaria clementii. And so the difference between these 2 flowers is actually in the number of cells in the nectar spur. And we can see from this analysis here which stage it is that occurs at. It's between the stages 2 and 3 in our graph. So when we look at the developing nectar spur, which starts as this tiny little outgrowth and then gets longer and then eventually becomes very long indeed, we can see that both cell division and cell expansion are necessary, but the thing that determines how long the spur ends up is how much cell division you have at this early stage. And so to understand how Linaria beckeri and Linaria clementii diversified from one another with those very different spur lengths so quickly, we need to now look at the genes that control cell division in the 2 to understand why they're different and why one of them is causing so much more cell division in the long spurred flower. So I hope I've shown you how we can use this evolutionary developmental biology approach, this Evo-Devo approach, to understand how the great diversity of flowering plants evolve and why we end up with so many species. I should finish by thanking my research group because, as I said when I showed you Erin and Mario, this work is never done by the person who's actually giving the talk. It's always done by a really dedicated and highly skilled team of people working in the lab. Of course, at the moment, they're not working in the lab. They're working at home, writing up their experiments, but I'm very grateful to them all anyway, and we'll look forward to having them all back in the lab again soon when the situation changes. I should also thank the various funding agencies, and I've put their logos around the outside of the slide. We're very lucky to have support from a number of funding agencies, including public funding agencies, to support the work we do in the lab. And finally, I want to end by saying that I'm very sad not to be able to be giving this talk to you today in the Botanic Garden. It's the most amazing place to work, the most amazing place to visit. But I hope you've enjoyed hearing a little bit about the research we do and how we use our living collection. And I do hope when times improve and we can reopen the garden to see you there visiting very soon. Thank you for listening today. Bye-bye.